we'll go into the discussion with an optimistic feel. The way I think we'll run this for the next 20 minutes or so, if I could just make a couple of introductory comments, and then perhaps I'll put one question to our panelists, and then open up the floor. I'd suggest that if you have specific comments of clarification, perhaps the speakers would be kind enough to wait around for 5 minutes to take individual discussions. And if you want to make a general comment from the floor or ask a general question, we'll take that after the panelists have said a few words. Listening to the discussion this morning and the 3 presentations we've had, I guess it's not so controversial that we will all agree we need the correct design to answer the particular scientific question that's of interest. Of course, this is complicated to some extent by what's ethical and what's feasible, what's practical, and that's difficult to discuss in general terms, the challenges that each particular situation brings to bear. I think we should focus in the discussion on confirmatory clinical trials. Of course, one of the difficulties is that you'll still be learning about your biomarker in many cases while you're trying to confirm the efficacy and safety of your actual medicine. But I hope we can focus the discussion on how to move forward in terms of the design and implementation of confirmatory clinical trials that involve a pharmacogenetic biomarker. And so the question for the panel at the end of this brief introduction would essentially be, what hurdles do you see to the wider implementation of a pharmacogenomic marker-driven confirmatory clinical trial? And I think actually the issues have fallen into perhaps 4 or 5 buckets, if I can use the vernacular. So the first would be, what standards for confirmatory clinical trials? Is it reasonable for regulators to apply all of the usual standards for confirmatory clinical development? And to take one example, the role of pre-specification, that all of the detail is put in the protocol and the statistical analysis plan and followed when the data come to light. And we could ask, for example, around how many independent validations are needed before a biomarker signature is used as a companion diagnostic in a confirmatory trial, or what evidence from early phase trials is required to support an enrichment strategy in confirmatory trials, and in particular, if you are learning about the biomarker and you only find out really critical information during the large sample that is the phase 3 trial, and of course that's not unexpected, how do you then make decisions and set hypotheses retrospectively? The second bucket might be, to what extent are adaptive designs a practical solution to this problem, either when we've got single markers or even more complicated when we've got multiple markers to consider, and what's the best way of finding out the relevant information in a timely way. And as I said at the end of Martin's talk, we've not just got to consider the methodological and technical challenge, but also the logistical challenges that are posed. The third question might be what to do in biomarker negative subjects, and here I'm using negative to mean those who are not of primary interest based on the hypothesis that we've got. How should biomarker negative subjects be studied in confirmatory clinical trials? And the starting position perhaps is that they should be included in some way unless absence of benefit or harm, absence of benefit or evidence of harm is already evident from the earlier phase work. And it might be interesting to see if anyone disagrees that biomarker negative subjects should still be studied if there's an open question. But what hypothesis to set and what level of evidence might you want to see in a biomarker negative population to follow Martin's talk, perhaps to make a claim in the whole population? And then the fourth question, to follow on from the talk of Dr. Devanarayan, is really how externally valid are the results of confirmatory clinical trials, given the variability, given what happens when you change from batch to batch, given what happens when you change from assay to assay. Are we making decisions on an evidence base which is externally valid? And so what we'll see out in the big wide world, so to say, can be reflected in the confirmatory trials. So those are the topics of interest that certainly that I took from the discussions and the presentations, but perhaps I could start with the panelists to get your view on the hurdles that need to be met before there's a wider implementation of marker-driven confirmatory clinical trials. And Emmanuel, perhaps I could start with you about adaptive designs, and you've got some experience in terms of the planning of and the implementation of adaptive designs, and it might be useful, I think, to rehearse some of your experience.
Thank you, Rob. Yeah, Emmanuel Zuber, heading the Food Development Biostatistics in Novartis Oncology. I was indeed involved in a trial as presented by Martin, and did face the different aspects of the preparation of the design. And maybe to start with the positive note, what I would like to emphasize that this type of design can actually answer one of the questions raised by Krishna on whether enrichment designs can allow some validation of the marker and also to gather some data on the utility of the marker in the general population, because you will really study the properties of your biomarker in your target population, in your confirmatory setting population, and you will gather data on patients who are biomarker negative as well as biomarker positive, so which will feed into this this data generation. Now, to implement those designs and to answer your question on the feasibility, I think it's important to consider that it very much depends on how much do you still need to learn versus the paramount concern that you still want to protect the integrity of your study and therefore to have a confirmatory study in the end. The more you need to learn, whether, for example, you have identified already the marker that you want to just confirm in your confirmatory study is one thing. Whether you are still fishing for which is the relevant marker, are those going to be one or two different markers? Is that a signature that you're trying to define? The more you go in that direction, the least feasible is your study. So Martin emphasized the fact that the methodological aspects from the statistical perspective are in place. I think the homework has been done there, but the practical hurdles to implement such a design are very high. So to quote a couple, in oncology, when your endpoints in a confirmatory study is a time to events like mortality for survival or progression-free survival, it takes time to observe your outcome while you are still recruiting patients. So the decision-making time that you need may require you to stop recruitment to have the chance to make your decision and then start again in recruiting, for example, in targeted population. That's one big hurdle, for example. So the disease setting where you are, the speed of recruitment that you can anticipate versus the speed at which you can observe your outcome is one of the hurdles. Another hurdle is the decision-making in such designs is a very complex process. It's often not possible to make it fully mechanical, just on one set of mechanical criteria, but you will want an assessment of risk-benefit, and therefore, who will make that decision? In a confirmatory study, you want an independent body to be involved here, an independent data monitoring committee, but how far can you let go of the decision capacity to such a committee when you're a sponsor are part of the things that need to be carefully investigated. Maybe a third and last hurdle is the time to actually set up such a study. Because of its complexity, you need to investigate a lot of scenarios. In Martin's slides, you had, for example, the impact of the prevalence of your marker, which does impact your operating characteristics of your study. There are many more scenarios that you need to investigate and to get full alignment on from the sponsor side and with the health authority. And all this process takes a lot of time. So maybe one thing to keep in mind is that I believe those designs allow a much better decision-making in terms of the quality of the information that you gather in your target population. yet the idea that those designs would really allow you to gain time is to be investigated on a case-by-case basis, because most of the time the setup time is lowering the overall development time impact of such an adaptive design, if that makes sense.
No, thanks for that. There really are different pros and cons to the different design choices, and Martin showed putting together the phase 2 and the phase 3 into one single trial, and of course you lose an element of replication, and you lose the time for decision making that you're talking about. So in some situations it will be a really strong design feature, and in other situations it will be a difficulty. Marie, your thoughts, please, on any of these topics, but in particular on what you see as the hurdle to a wider implementation
of adaptive design. You mean? Yeah.
Of anything to do with confirmatory trials for pharmacogenomics.
Okay. I'm Marie Watissee. I'm Associate Biostatistics Director for Shire Pharmaceuticals based in the UK for specialty pharma. And to answer your question, all the hurdles you listed, Emmanuel, are the one that I identify for any kind of adaptive design trial. The main issue I see here is that when we talk about learning and confirming for a biomarker, and especially for pharmacogenomic marker, what I would call learning is actually identifying the biomarker. So you're, in practice, I would think that in your phase 2, you might genotype for maybe a couple of genes and have 10, 20 SNPs that you're looking at. And that's to me the hurdle for such an adaptive design, because in your learning phase, when you're trying those SNPs and testing for association with treatment, you're likely to analyze in batches to actually reduce your variability, because you can account for those for the technical bias. But then in the confirmatory phase, if you're lucky enough to have identified a marker, then your marker is used for the randomization. So you need to have a test that is, you can't afford to wait 3 months for the wet lab and the DNA to be extracted. So that all the timelines and practical issues are to me the main thing.
Thank you. And Mathias, any thoughts from yourselves on these hurdles that we've been discussing? Perhaps you even have some solutions first.
Mathias Chiano, actually, from GSK in Stevenage. I mean, this is actually quite challenging. I don't know actually whether I have a solution. There are things actually that we ponder about, and we really we're hoping actually that maybe the agency will have some proposals here actually for us to take home. I mean, one of the very practical challenges actually is particularly to do with genetic biomarkers is the fact that we conduct trials actually in diverse populations, and genetic variability actually tend to be, to differ across populations as well. And when you come to test or to do the learning, it's always actually quite desirable to look at focus populations actually where they're fairly homogeneous in a way, so that you have actually the remote chance of identifying a biomarker. And to validate that, actually, you need to, in a confirmatory way, you probably will want to try that out actually in a follow-up population actually that is very similar to your learning population. So, no clear answer here, I don't think.
It's all right, there's still a day and a half left. So, opening up to the floor then on the question of methodology for confirmatory clinical trials, some comments from the floor, please, on the topics that we've mentioned or on other hurdles that we have not mentioned, and as I say, solutions welcome. Yes, please.
My name is Ingolf Kaskorbi. I'm from the University of Kiel in Germany in the healthcare professional group. I think when we talk about biomarkers, we have different levels. If we are using a quantitative trait or just a qualitative trait like a genetic marker. So that's the first hurdle. If you speak from confirmation or validation of a marker, I would expect that this is the first step in the quantitative trait to have the right methodology. And after that, you can use it in the study. So my question is, when confirming studies, was there always the same methodology behind it, or was it just a different set of patients? I think that is the first question, if you have to confirm a study, if you are using the same methodology.
Seems logical. Does anyone want to make a comment on that?
As far as study design goes, it shouldn't.
Use mic, thanks.
As far as study design goes, it shouldn't matter. But as far as evaluation goes and all other considerations we discussed, it certainly it depends on a case-by-case basis, whether it's a quantitative expression marker or a qualitative genetic marker. There are unique considerations for those kinds of markers. But study design shouldn't be very different, as far as I know.
Thanks. There's a question in the corner.
Thanks, Adrián Llerena from Spain. So just one question. We are sometimes assuming a therapeutic effect from pharmacological effect. So my question is concerning about biomarkers of placebo or nocebo effect. Take depression as a typical example. So we are looking for biomarkers of the drug involved in the response to antidepressants. But all of us, we know that 40% of the response will not be related to the drug. So how can we take this situation into consideration? And second, part of the biomarkers that we call pharmacogenetics biomarkers could be involved in the vulnerability to the disease. So how can we stratify the analysis in order to take this into account? Sorry to be provocative, but this is one of the main issues that worries me. Thanks.
Sorry, I'm not sure I understood the second part. Can you repeat the second part, please?
Yeah, some of the biomarkers that we are called pharmacogenetics biomarkers. If you want, I can give you an example. CYP36 for us is a typical classical biomarkers, but there are many references connecting CYP36 with vulnerability of psychopathology. Well, many groups, as myself, we are working on the relationship with psychopathology, suicide, etc. So when we are testing biomarkers just in a disease, how can we stratify, considering that this biomarker could be at the time related with the vulnerability of the disorder? So we should stratify volunteers or patients according to that. Is that clear now, or is it a bit confused?
Let me start while people have a think. I mean, on the second point, we've had some general discussions in the regulatory system about how to choose to stratify a clinical trial, and obviously you're looking at trying to identify factors of prognostic and predictive influence. So both types, if you like, in terms of the disease characteristics. It might be the region, it might be demographic characteristics, it might be due to the pharmacology or the mechanism of the drug, and to try and identify those factors which are key. So regardless of whether they're prognostic, affecting the vulnerability and the outcome for the individual patients, or whether they might be predictive in terms of response. See if anyone on the panel wants to have a go. Martin?
Yeah, I think that in principle, it's a quite different thing if you look at just prognostic biomarkers, and they can probably be using the standard methodology in clinical trials, as Rob mentioned, with biostratification dealt with, and they probably also do not play such a big impact in the for the regulators, because it doesn't say anything about the effect of the drug itself. So I think the really crucial issue is the predictive biomarker, if you can really identify the patients for which the drug works. Obviously, there may be biomarkers which are both predictive and prognostic, and then of course they again become essential, and all the complexities that we discussed come up.
And does anyone want to try and tackle the first question of identifying placebo responders in depression? There's a challenge. Oh, yes, please. Yeah.
On the other question about predictive versus prognostic, practically, predictive is more interesting than prognostic. If we are, because in most cases we are looking to differentiate treatments. How our treatment, the candidate treatment, is better than the benchmark reference standard. And so if we can not only predict which patients are going to respond to our treatment, but also predict the patients that this signature will only have an effect, will only show the responders that are specific to our treatment, and will not show a similar differentiation for the other treatment, that will be ideal. So having both a prognostic and more importantly a predictive component to it that is specific to our treatment will be of more interest, I think. And I think the slide that Krishna had for one of the examples where I think clearly illustrates what I was saying now. The signature is differentiating the responders and non-responders for the treatment of interest, but it doesn't have a similar predictive ability for the other treatment. So, and other question was about
The other question was, if I understood, around the work to identify placebo responders, essentially in conditions like depression.
Sorry, no, pharmacogenetics of placebo effect. Don't take the sample. Yeah, you can take in general if you want.
The pharmacogenetics of a placebo effect.
Are you asking this in the context of being able to identify patients that are more likely to have a placebo effect? Okay, so whether it's pharmacogenetics or proteomic or genomic, whatever the biomarker opportunity is, we clearly need some access to cohorts of patients that have been tracked longitudinally. And so if we define placebo effect as, for instance, patients getting better over time, right, on their own, for whatever reason, like in Alzheimer's, also in depression, the placebo group that's treated versus a treated group over 6 months, some of the placebo subjects may actually show a positive response, and some may have a negative response. And when you say placebo effect, you mean those subjects that are automatically showing an improvement, right, just on placebo. So for that purpose, we certainly need access to a longitudinal cohort where these genetic and genomic and other kinds of biomarkers have been measured at baseline. And if we're able to identify a signature with a single biomarker, a single genetic marker, a combination of markers that predict which patients are likely to progress in the disease and which patients will not progress, actually will get better or not progress in the disease, and then can apply that signature in a clinical trial and look for and enroll patients that we think are more likely to progress in the disease based on the genetic or other biomarker.
Thanks. It's a general comment from a number of these workshops. We often end up talking about disease models and sharing placebo data across pharmaceutical companies. Yes, please. Could you use the microphone, please?
Thank you. Tor Vergata University and St. Peter Hospital in Rome. It's just a short comment about prognostic markers and the impact that they may have on pharmacogenetics. A PCSK9 study by Ellen Obst in the United States on familial hyperlipidemia, where she found a missense mutation knocking down the gene and lowering LDL in patients protected from the phenotype. Well, that has, in my view, an impact on pharmacogenetics. Number one, because it's very easy, or is easier to knock down a gene rather than to boost its function or increase its function. And the other thing is that statins have an effect on the gene, raising the transcriptional level of the gene. So turns out that in terms of LDL levels, statins are not so good. So I would say that that's an example where a prognostic marker for a metabolic phenotype also has an impact on pharmacogenetics, and certainly raised a lot of interest in the pharmaceutical industry, exactly because it is easier to abolish the function of a gene rather than to boost that gene up. So it's one good example that comes to my mind.
Thanks for the comment. Is there any final burning comments to be made generally please. Short comments, please.
Okay, Cynthia Song from Health Sciences Authority of Singapore. There is a fairly rigorous statistical framework in population pharmacokinetics for when you could allow an intrinsic a factor to be used to put into labeling. I guess this is more a comment for the biostatisticians. Why don't we leverage on that and just use a marker, a genetic marker, as just yet another intrinsic factor?
I think it's a good comment, and there's a lot of literature about adaptive designs, and there's a lot of literature about subgroups, and of course there's an interplay between all of what we're discussing and the wider discussions of those topics. Others? Yes, please. Short, please.
Yes, it's more of a question over a question. Regarding your first bullet point, how would you answer that question if you were referring to a diagnostic instead of a therapeutic?
I think that's on the agenda for tomorrow. I can't imagine in the minus 3 minutes we've got left. I think that's too big a question, but thank you.